Am I audible? Okay. Yeah, thanks to Kavita and Deepa for organizing me. And it's just the beginning of the school. So what I'll actually try to do today is sort of, I would have wished Bruce would have finished some rest of his lecture because that would have given some basis to what I was going to talk about today. But what we are interested in our lab is to actually look at complex traits in yeast as a model and try to understand some of the fundamental properties of how genes and environment interact to bring about changes in the phenotype. And what I'll try to do today is to sort of try to press the fact that phenotypic robustness, which I'll describe in a bit, is the fundamental basis of how genetics of complex traits is regulated. Okay, so consider a population which sort of is distributed, like has evolved and has a particular distribution. If a new mutation actually takes place in this, what is either by any process, by drift or by mutations, usually that is purified by selection. However, what would happen is that if all mutations are purified, then the population would not be able to adapt if a new optimal fitness comes. This population, therefore, would not survive if the fitness paradigm has changed. But what we know from various studies is that this is not true. So that means the populations are able to actually also adapt to the new fitness by either changing their distribution of their distribution, where the new environment allows the population for the network that is regulating the fitness here, allows it to be expressed here, or the regulators which are sort of holding this network together are altered, and therefore this network is now not one sense buffered and it becomes unbuffered. And this property where the phenotype is under a buffered state, and when it sort of moves to a novel environment, it releases genetic variation, is sort of defined as phenotypic. So what it allows us to sort of see is that there are several variants which are under buffered state in this population, but when either the environment or the regulator changes, these mutations actually can express themselves and bring about a change in which we now term, which these variants are actually termed as cryptic genetic variants, and because they are not showing their effect in this initial population, but only when they are either in alternate environment or regulator. This property has importance for adaptation because populations have to balance between buffered and revealed states, and eventually these adaptations lead to speciation and generation of new phenotypes, because when in either of these states the new variants that are now getting expressed can actually generate new phenotypes which can lead to speciation. So in terms of how these cryptic genetic variants, which are sort of hidden in terms of their expression of phenotype, are so could be that, or so it could be either of them. It could be the lower or higher state, or could be completely new different environment which now has never been experienced by the organism. So it could be either of them. So think in terms of how people have been sort of trying to define cryptic genetic variation is that, let's assume that in a population there is a disease allele which is under a regulator, a network of genes which is sort of regulated by a regulator, and there is a particular disease allele. Now in this population the disease allele is not showing its phenotype. So that means the disease alleles effect of phenotype is buffered. If there is a change in either the environment, which could be, say, for example, metabolic environment for a particular human population, or mutations in the regulator, which now allow the buffering of the phenotypic buffering of this disease allele to be lost, what would happen is that the population would be now, the effect, phenotypic effect of this disease allele would now be visible in the population as a disease phenotype. So what I want to stress over here is that these cryptic genetic variants, which are sort of buffered here, have the ability to, if they are buffered, they don't show a phenotype, but they have the ability in an unbuffered state to show a different phenotype. Here in this case, a loss of buffering leading to a disease phenotype. And therefore, if one were to do mapping studies in these populations, even though the disease allele is present in both the population, the control and the disease population, you would not probably identify these disease alleles as causal alleles for the disease because they are present in both. Only reason why this disease allele is now a causal agent for disease is because it's in an unbuffered state. So it becomes very important for us to actually understand the genetics of phenotypic robustness for us to comprehensively understand what are the causal disease alleles which lead to disease phenotypes. And what is the, so here the buffered state means that the network of genes which is regulating a phenotype, they are under sort of a regulator which doesn't allow the mutations which are present in the population to be expressed as novel phenotype. So there have been several studies in past decade and a half where people have tried to understand genetics of various complex diseases, and one of the questions that always comes up is that there is, even though we know several of these variants, we still cannot account for the large proportion of the variation in the population, and that's sort of termed as the missing heritability. And this could be one of the reasons why we are not able to identify many of the disease alleles, because they are probably under a buffered state in individuals, whereas in disease only they show their effect. Okay, so people, is it not that we are the first ones to sort of propose that phenotypic robustness is something that is important for evolution and adaptation? People have been studying this property for a very long time, and flies, Drosophila models, and Arabidopsis, people have actually done evolutionary studies where they have actually evolved. They have taken two different evolved populations, done mutations in them, and looked at whether the populations lose their phenotypic robustness or not. And depending on which population does lose its robustness, they term that this population is now become unbuffered, whereas the other population hasn't. The only molecular gene which sort of has been implicated in phenotypic robustness is HSP90 from Susan Lindquist's lab, where they propose that HSP90 being a protein chaperone is both, yeah. So when you say, so robust phenotype is that it is all the variants that are in the population, they do not show their phenotype. So that means their phenotypes are sort of buffered in sense. So the total variation in the population is very tight. When you lose robustness, that means either the central regulator which was controlling those variants, or the environment in which now the regulator cannot function, does not allow the population. So now what happens is that the variants which were previously phenotypically silent now are able to show their different effects. Therefore, now you will see a larger variety of phenotypes, a larger range of phenotypes from much lower to much higher. Yeah. uh, we don't know very much about what are these regulators. So that's why I'm trying to say, like, there have been very few studies which, so the question was that, is there a one-to-one mapping of the regulator and the effector gene that the regulator is regulating, right? Is that what the question is? I'll come to that towards the end of my talk. Okay, that's a good question. Yeah. Mhm. Yeah. So canalization and robustness are sort of interchangeably sort of used in one sense. So when I'm using robustness, it's sort of phenotypic canalization as well in one sense. So HSP90, as I was saying, is the only known molecular gene, functional gene, which is implicated for phenotypic robustness. And that is because it is a protein chaperone uh, which the function of that chaperone is to take misfolded proteins and do proper folding so that the functionality of the protein is not altered. So if you now can mutate or disturb the function of HSP90, the chaperone, you suddenly start seeing a variety of effects because of the fact that the misfolding protein response is now downregulated. So it's both termed as a potentiator, because now if you can reduce the activity of HSP90, you can actually potentiate changes in phenotypic robustness, or as a capacitor, because now it can actually hold a lot of variance in a tightly regulated funnel and just allow a certain functional component of those variants to be expressed. People have also done indirect studies and have inferred that phenotypic robustness should be something that is present in populations. We know from a lot of high throughput gene interaction studies, protein-protein interaction studies, and transcriptome studies that there is a redundancy in the genetic pathways within a cell. We also know that there is a high amount of epistasis in most of the complex traits, where a quantitative trait locus identified in one population usually does not have the same effect in the other population. So, and the interactions between two quantitative trait loci could be very different in different populations. So all of these studies actually do indicate and try to understand the mechanism, the genetic mechanism, as well as the molecular mechanisms of what is phenotypic robustness. Why is it so difficult as compared to, say, mapping variants for quantitative traits or diseases or like any other complex trait? Is because you have to do genetic perturbations in a large heterogeneous population to see its effect. It's unlike taking, say, a yeast deletion collection and saying what happens if I delete this gene and let's look at the phenotype. And because the single genes usually do not, because of the genetic redundancy, single genes usually do not change too much in the terms of phenotypic robustness. So this has been a challenge in the field, and we sort of try to look at this problem in yeast. But before I go there, I just would give a very, very brief one slide primer, and I am sure there would be people here who would explain this in much more gory details of how this is done. So standard QTL mapping is mapping for, so you take a single marker, you ask whether the population, it's a biallelic marker in a population, and you're asking whether the population which now divides based on this particular alleles of this particular marker, whether it is significantly different for the mean, assuming the variances are equal. So this is what the standard QTL mapping does. These are the two variances of the two alleles, two loss alleles of a single marker, and the variances are equal, and these if the difference in mean is significant, you term that as a quantitative trait locus. Now, what was also realized and very recently, and this I'll just explain in like the next slide, is that it not always need to be that the marker is changing just the difference in mean. It can also change the difference in variance. So you can have two kinds of comparisons where a marker doesn't change the mean at all, but only the variance, or the marker changes the variance as well as the mean. So there is a directionality in which the variance is leading to. So if you now do QTL mapping, you can do QTL mapping for all the three kinds of markers. Markers which only affect the mean, markers which only affect the variance, and markers which affect both mean and the variance. And so one of the ways to sort of do this is to, in a very clean way, is to look at the biparental populations. And why? Because biparental populations is derived from a single cross, and you have a large heterogeneous segregating population. And you can actually study effect of single variant across all the individuals in the population. These variants are not artificially induced. They are variants which are naturally derived and have evolved based on the evolutionary history of the parents that you are studying. And because now you can actually compare each variant, you can actually do comparisons of robustness mechanisms that these diverse strains must have gone through and how they would have buffered their genetic networks to bring about the changes in whatever environments they were actually evolving in. So to do that, we sort of used a concept which was recently actually proposed by Ronengard and Wilder in 2011, where they proposed using variance QTL mapping as a method to identify variants which have the genetic basis but are beyond the mean effects only. So this was like not just doing mean QTL mapping, but doing variance QTL mapping as well. And because of the fact that the genetic variation, the fact that you see a larger variance for a particular allele, as I was showing you in this slide, because now there is a change in the variance, the subsequent studies propose the idea that this change in the variance could be because the allele has differential genetic interactions, which now leads to a wider variance. So this variance QTL mapping not only allows you to identify loci which not just affect mean but also variance, and give a very strong indication of what are the genetic interactions which sort of are being regulated by these QTLs. And so we used this method to actually interpret robustness. And how we did that, so our hypothesis was, or our interpretations was, that a mean QTL, which only affect the mean, shift the mean only while maintaining robustness. So that means that because the variance is not changing, so the robustness is remaining intact, only changes in the mean. The variance QTL, one allele keeps the robust phenotype, which is this red allele indicated here. The blue allele actually loses robustness. So therefore, one allele is robust, the other allele leads to decanalization or loss of robustness. And in the third scenario, which is combination of both of these, one allele leads to loss of robustness and is accompanied by a shift in mean, and that shift in mean can be either positive, in the positive direction, or in the negative direction. So to do this kind of analysis, we actually looked at yeast as a model. Now yeast is a very wonderful model for doing quantitative genetics because it can exist as a haploid state. So you can actually right from a diploid heterozygous state to a haploid state, to take two haploids, cross them and make them diploid, and then do the segregants, which again would be haploid. So your allele frequencies are they're always biallelic, so your population divides equally into two halves approximately. So what we wanted to address? We wanted to address, we knew from previous studies that specific example of gene-gene interactions do exist, but there was nothing common that was coming out of these VQTL studies or variance QTL studies. And what was actually more prevalent in the field was that the variance QTLs are usually a subset of mean QTLs. They do not, and they have very small effects, and they do not really have a role to, the current evidence did not show a strong reason for them to play a big role in adaptation and speciation. So the confusion was that are the variance QTLs just a something that we would detect when we do variance mapping, or they really do have a specific role to play. So the questions we wanted to ask was that, does regulation of phenotypic robustness play a significant role in genetic architecture of these of the quantitative traits, and is loss of robustness good or bad for the population? Does it, like, if a population is robust, like it's buffered, and suddenly there is a loss of robustness, does it always lead to an advantage, or is it a disadvantage? And what is the actual scenario? To do that, we actually, as I said, we used yeast as a model. In this case, this was a data set from Leonid Kruglik's lab, published in 2013, where they took two strains which were evolutionary very divergent. This is a lab strain, BY, which was the first eukaryote to be sequenced. Evolved in lab for past 100 years. Prior to its lab avatar, it was a isolate from fig. This is a vineyard isolate that was isolated very recently from a vineyard in California. They crossed them. So these were two haploids. You cross them, you get a diploid, they let them undergo meiosis. They isolated about 1,000 segregates, individuals from this cross, genotyped all these 1,000 for 11,000 markers, and phenotyped in 30 or like 40, but we considered only 30 environments for colony size variation. Now, colony size is a growth phenotype and is a typical yeast fitness phenotype. So if the growth is robust, if the colony is bigger, the growth is robust. If the colony is small, the growth is not robust, or growth is poor. So it's a direct correlation to the fitness. So we took this and we did the 3-QTL mapping for all the 11,000 markers across all these 1,000 segregants for all the three kinds: mean QTL, variance QTL, and mean and variance QTL together. And this is what the distribution was. Obviously, this was a published data set, so they had already done mean QTL analysis, and that's what we also found. We found that majority, about 70% of the variance of the QTLs that we identified were mean QTL only. Very small proportion, 4%, were just the QTLs which changed just the variance, not the mean. But a significantly 1/4 of the total QTLs that we identified were the QTLs which not only changed the mean but also changed the variance. And now these were the very interesting ones, because they now have, these were the alleles which had the ability to change the mean but also change the phenotypic robustness of a population. And so if you now look at, so these are the various environments that we considered. If you look at the color codes, black is where both of the black is this, red is this one, and blue is this QTL. What you can see is that there is a distribution of, based on the environment, you can actually see that certain environments, only the mean QTL is the QTLs that are explaining the variance for that environment. In certain other environments, only the QTL, or what we term as both QTL mapping, was able to explain this, or a significant proportion of the QTL, VQTL variants were able to explain variation in these set of environments, and these are majority mean QTL. So what it sort of showed us was that the variance QTLs were being, the variance QTL and VQTL, which are the both category, were being identified as mean QTL, but they were also now, even though we were studying them as mean QTL, they had the ability to change the robustness of the population. So we had been studying these QTLs, or the original paper did study them, but they were not looking at robustness. So now the question became was, is this, now that we know that there are these environments in which both of these mean and variance QTL, the variants have both a mean and variance QTL, properties. Is the release now, because now there is a loss of robustness, is it good or bad for the population? So this is how we sort of, if I'll just walk you through this one of these graphs, and that's exactly what it is. So this is the marker, this is chromosome 12, this is the marker position, this is the environment, 4NQ, which is a DNA damaging agent. Now, and on the x-axis is the sort of standardized phenotype. The blue is one allele of this marker, BY allele. The red is the RM allele. So there were two parents, BY and RM. And this is the frequency in the population. So as you can see, in this case, release of variance, which is indicated by this flatter curve, leads to a log reduction in the mean. So that means release of variance is detrimental for the population. In this case, the variance is not very different. So this is a typical mean QTL, and there is no difference in variance. And in magnesium sulfate, for this marker, you can see that the release of variance, which is indicated by this long tail of the blue curve, increases the fitness of the population. So what we were able to see was that the certain variants increase the mean value by releasing variance. In certain variants, it decreased the mean value. And however, we were not able to ascertain any particular pattern in terms of whether there was a certain category of mutants or variants which were always beneficial or always detrimental, till we actually looked at individual environments. And so what we did was, we did a simple correlation analysis between the mean and the variance for all the QTLs in a particular environment. So if it is a negative mean, that means generally all the variants lead to poorer fitness phenotype. So as you can see from this figure, these are all the environments that we consider. There was a category of environments in which generally the loss of variance, or like loss of robustness, led to negative fitness. Then there was a category in which loss of fitness led to a beneficial effect. And there were very few categories in which loss of fitness was not poor or good. So what the take-home message from this graph is that it is the environment which, or the evolutionary history and the environment, which determines whether loss of robustness would be beneficial or detrimental for a population, not the environment per se. Because if you can read through these environments, there are several environments which are sort of both DNA damaging, both carbon sources and all those. So it's the environment and the evolutionary history which sort of determines what would be the scenario for a given variant. Okay, so one of the, when you have environments, when you have sort of genetic variants which can contribute to differences in mean or variance, one of the properties of quantitative traits is gene-environment interactions. That means an environment, genes and environment interact to determine whether it would be a beneficial effect or a detrimental effect. And one of the classical ways of looking at it is like conditional neutrality, where a single variant has, a particular variant has no effect in one environment, but it hugely affects the fitness in the other environment. The other two classes are scale effect, where in environment 1, the magnitude of difference between the two variants, blue and the red alleles of a given variant, has lesser magnitude than in environment 2, or more interestingly, antagonistic, where the one variant is better in one environment, the other variant is better in the other environment. Now, this is how most of the, and these are called reaction norms. So this is how this sort of lines are called reaction norms, and people have done, and we also did gene-environment interaction mapping for the same data set prior to looking at it at robustness level. So what we wanted to see was, can we now use gene-environment interaction mapping to infer about phenotypic robustness? And this is how we wanted to infer it. A scale interaction is loss of robustness, but the shift in the population mean is, so loss of robustness shifts the population mean in the same direction, but only the magnitude is different. Whereas antagonism occurs when there is a loss of robustness, but the mean has an opposite effect. So what we are trying to sort of infer is that antagonism not always need to mean that the mean has completely, a population has moved from a negative mean to a positive mean or a positive to negative mean to bring about this effect, but it could be just a consequence of loss of robustness. And we actually saw those kind of examples. For example, if you again walk you through the graph here, this before I walk, so we compared two environments, paraquat, which is a oxidative agent, and copper. In paraquat, the mean of BY containing individuals is less than the mean of the RM individuals for marker on chromosome 13 of this. In copper, reverse is true, where BY is better than RM. Now, this is a plot of fitness profiles of all the individuals containing BY allele. This is the growth of these individuals, standard phenotype of these individuals in paraquat, and this is in copper. And as you can see, majority of the individuals actually are better in copper, but poorer in paraquat. And these are the various variances and means of this. So according to antagonistic interaction of pleiotropy, one would expect that all these individuals should have to bring about this change in change in the mean. One would expect that these individuals should move to this quadrant to bring about this change. So this is exactly what I'm trying to show schematically here, where all the individuals here now need to move to this quadrant to bring about a change in the mean. But what we actually see is this, that the population doesn't really move that much. It's the loss of robustness or release of variance which actually leads to the change in mean. Because if you now compare variance of BY allele in copper, this is 0.52, here is 1.26, and even though the paraquat variance doesn't change much, the mean has changed from positive 3.4 to negative 3.3, and from negative 0.2 to 0.2 here. So what is happening here is that just because now in presence of RM allele, the robustness is lost, and there's an increase in variance, the mean has shifted from this being better here to this being better here. So what it explains is that the robustness or the loss of robustness could be one of the mechanisms through which antagonistic interaction which we see in gene environment interactions could be brought about, rather than just a complete flip in the effect of that particular mean, particular variance. No questions? Okay. Okay. So you are understanding everything I'm saying. So right now, what we were sort of interested was, we saw that the variance were the loss of robustness or increase in variance was something that was based on the environment. We wanted to also understand, is it genetic in nature, or is it something that is, because in gene environment interactions one needs to be very careful about whether the effects that you're seeing are genetic or mostly environmental. So we wanted to see whether this release in variance is genetic nature, and whether across two environments same variance cause the effect, or do they interact with, there are different interactions that happen, genetic interactions that happen to bring about a change in variance. So we used what is called TWO-QTL mapping, in which rather than, so all the previous examples were like a single marker divided into sort of two classes. In this mapping, you actually have a 2 by 2 matrix where you have 4 classes. You divide your population into 4 classes and do all the two-way comparisons to understand which of the combination actually leads to loss of variance or not. So we took So for example, we have, if you consider growth in galactose versus growth in 4-HBA, hydroxybenzaldehyde, for two alleles of this marker, you can see that the BY allele has a higher variance compared to the RM allele, and that is also indicated by the higher variance values of 1.24 here. Now, what we were able to show was that this uh, increase in variance of BY allele is due to different genetic interactions in two different environments. For example, this marker, which is the same one, interacted with marker on chromosome 13, and the BY allele interacted with marker chromosome 13 to bring about a change in hydroxybenzaldehyde, which is this axis, and you can actually separate the effects of the two. So the total variance is wider compared to when you have just the RM marker. Similarly, in this again marker, same marker, interacted with the marker in chromosome 15 in galactose, where the BY allele again sort of changed the variance compared to, had a higher variance compared to the RM allele of this marker, which sort of indicates the increase in variance in this direction. So what it shows is that if a particular marker which is changing the phenotypic variance, it is able to interact with different other genetic markers in different environments to bring about a change in the variance for that particular environment. And we compared this across variety, like all the QTLs that we had mapped, and we identified that these markers were actually very highly enriched for gene-gene interactions. About 57% of the markers were which were originally identified as QTL and VQTL only were actually showing genetic interactions. And the number of, one could classify these interactions into two categories. One were these environment-specific hubs. So these made hubs where a particular marker on chromosome 2 in a given environment interacted with several different markers on 4 different chromosomes, actually 5 different chromosomes, to bring about a change in variance. Or you could have markers which are common across environments. For example, a marker on chromosome 14, which each of different color of the this line indicates different environments. So you can see that this marker is actually pleiotropic across multiple environments. So we could sort of, this indicated that the environments, these markers formed a genetic network where these certain loci were hubs which regulated variance across either multiple environments or within an environment. And rest of the gray lines are other examples which I haven't detailed here. So what it sort of indicates is that while there is, we know, so for example, we know from previous studies, several studies that have been published about gene-gene interaction, protein-protein interaction, that there is genetic redundancy. So what we wanted to ask was that, is there genetic redundancy even in the markers or the variants which regulate phenotypic robustness or not? So one of the classical example, as I told you earlier, was HSP90, but there has been a problem in just having one example which, or one drive or one hub which sort of regulates robustness, because robustness is a very important property of for a population, and it's very unlikely there would be one. People have proposed several other ideas, and they have sort of indicated that there should be several more robustness molecular genes, but they haven't really identified any sort of genetic basis for it. And previous VQTL studies have really not been able to actually ascertain the genetic basis of this genetic network of regulating robustness. So what we did was, we actually, as I said, we did 2-QTL mapping, and we did rather than just doing 2-QTL mapping, which is sort of commonly done in the field where you take 2 mean QTLs and do 2-QTL mapping or 3-QTL mapping, we did 2-VQTL mapping, where we took 2 variance QTLs and asked whether these 2 variance QTLs now genetically interact to change robustness or not. So let me walk you through this. This is the same population divided into 4 categories: BB, BR, RB, RR, for the order given here, chromosome 3 marker and chromosome 8 marker. And you can see while these 3 combinations have pretty much same mean values and variances as sort of indicated by here, only this particular combination of BB, BY allele of these 2 markers leads to a significant increase in variance, and that is sort of shown by the spread here. Now this is chromosome 3 interacting with chromosome 8. So we know that chromosome 3 variance QTL interacts specifically, BY allele of chromosome 3 interacts specifically with BY allele of chromosome 8 to bring about a change in variance, whereas rest of the combinations don't. And we were also able to see that this is not the only case. Chromosome 3 actually interacts with several other variance QTL markers to bring about a change, which you can see here, where almost in all the cases, chromosome 3 here interacts with chromosome 4, specifically with this combination, chromosome 3 here with 14 in this combination, 4 with 8, 4 with 14, and 8 with 50. So this sort of becomes a genetic network which is actually holding the phenotypic robustness in place. And a specific loss of specific combinations of alleles leads to loss of phenotypic robustness. So what we were able to identify, not just this, there were several others which are sort of the genetic networks which regulate phenotypic robustness and interact with each other to maintain phenotypic robustness. And this sort of is an idea that I'll show you as a model that we want to propose, how genetic robustness, phenotypic robustness is sort of fundamental to various complex traits. So what I've shown you today is that we were able to reproduce, as I said, like VQTL mapping is not something new. Mean QTL mapping was already done in the population, so we were able to identify most of those markers. And we were able to explain whatever the variance that was there in the population. However, we were able to identify that in at least 30% of the statement that I made, that these markers, these variance QTL markers actually interact to make a hub which is regulating variance, sort of makes us propose this hypothesis where we propose that phenotypic robustness, which is genetically regulated, is the primary network that regulates complex traits. Now consider a network of these causal variants which are regulated by these regulators, which is indicated by these squares, which are the robustness regulators. If these regulators are present, the effect of these variants would not be visible in the population. If there is, under most common conditions, these regulators are able to maintain buffering. So that means the population is able to maintain its phenotype without a dramatic change in even though there is a change in the environment, they are able to maintain phenotype. That means the population is robust. And what we do identify when we compare these two populations is that there are these QTLs which now become causal, because they are the ones that are explaining the variance between a buffered and a slightly unbuffered population. Only under very specific rare instances, which we were able to identify because we did a large number of environments, you see a case where even the hubs which are regulating the buffering are lost, their functionality is lost, and therefore every other QTL is visible, and this loss of buffering leads to a completely diversified variants showing novel phenotypes, and this happens in a very, very rare instance, and that's what the idea is. So what we sort of think happens is that if one wants to understand the genetic basis of phenotypic robustness and map variants which account for this, one would have to actually map a large number of environments. Previous studies, for example, at least in yeast and several in Arabidopsis, where people have tried to look for it, have looked at very small number of environments. For example, in yeast, most analysis is done in different carbon sources. We found that none of the carbon sources actually have VQTLs. They're all mean QTLs. They're not variance QTLs at all. In Arabidopsis, the environments that they considered were only 4 or 5, which were not large enough, even though the population sizes were large, but the environments were not large enough or dramatically different enough to bring about an effect of loss of buffering. So this sort of brings back to the first slide I was saying, that in this buffered state where a disease allele is present in the population, if the buffering of this network is lost, that means a state in which the population is in the adapted state, only when the buffering is lost, be it with a change in environment, that means change in diet or nutrition for human populations, or new mutations that can actually arise in the regulators which are regulating the phenotypic robustness, you see a maladapted state where a certain proportion of the population starts expressing the disease phenotype. And this is what we think. Therefore, we think that understanding phenotypic robustness becomes crucial to understand whether a particular disease variant would show its effect in a particular individual of a population or not, because the network that might be regulating the expression of that variant would be completely buffered. Therefore, we would not see its effect in the population. And finally, so I'd like to end here. So most of the work was done by Anupama, who's here in the audience today, and a computer scientist who got interested in coding for biology, and funding by TFR. This paper we had sort of written up. It's published on bioRxiv right now. We are doing some iterative editing to sort of think about it in a different way. And your inputs maybe after this talk would be very useful to sort of, if not to me, then to the first author Anupama, to sort of tell us if there are any other things that can sort of concepts that can be added to this body of work to sort of make it more plausible and more sort of useful for the genetic audience out there. So thank you for your attention, and